Welcome to the deep dive. Imagine trying to track something that's constantly changing shape, adapting all the time. That's kind of what scientists face with viruses. They're just incredible at evolving, diversifying,
Mm.
and understanding this rapid evolution. It's like trying to hit a moving target, isn't it? It's
Mm.
so critical for, well, everything from pandemic preparedness to finding treatments.
Exactly. And the sheer genetic diversity of viruses globally means if we don't have precise, accurate ways to analyze their genomes, we're basically flying blind.
Flying blind, yeah.
Accurate reconstruction isn't just about saying, okay, this is virus X. It's about getting a true biological picture of individual genomes and, importantly, all the different forms, the quasi-species or variants that might be lurking within even a single infection.
Right, all those little variations matter.
They fundamentally matter for understanding impact.
So today, we're taking a deep dive into some fascinating new research. It's centered on something called BonoboFlow, which is all about viral genome assembly and haplotype reconstruction using nanopore sequencing reads. Now, this isn't just another technical paper. It really promises to be, well, a bit of a game changer in how we understand these microscopic shapeshifters,
That's the hope.
offering a powerful shortcut, maybe, to being genuinely well-informed about viral genetics. So our mission today, explore the big challenges in sequencing viral genomes and see how BonoboFlow aims to tackle them. So let's set the stage first. What's the ultimate goal here when we talk about understanding viruses genetically?
The goal, well, simply put, is building accurate, detailed genetic maps. Ride maps, if you like, for these tiny invaders.
Okay.
Without those maps, trying to make critical public health decisions or design effective drugs,
Okay.
it's like navigating with a really blurry, incomplete guide.
The huge risk.
That's the risk we faced, yeah, especially with some of the older methods.
So how did we start trying to map these things? What were the first hurdles?
Well, the journey really began with traditional Sanger sequencing. In its day, it was reliable,
Okay.
but it was expensive. It had limited sequencing depth, meaning it couldn't read very far into the genome in one go.
Ah.
And low throughput. You couldn't process many samples quickly. So for large-scale studies trying to track viruses evolving across populations, it was just impractical.
So it worked, but slow, costly,
Yeah.
couldn't keep up with the sheer scale of viral diversity.
Precisely. Then came next-generation sequencing, NGS, platforms like Illumina.
Big step up.
Oh, a massive leap in throughput and depth. Suddenly, we could generate huge amounts of data very quickly.
Okay.
But, and this is where it gets interesting, NGS brought its own set of problems.
Always a catch, isn't there?
Seems so. The main one was genome fragmentation. NGS basically shreds the viral genome into loads of very, very short pieces.
Okay, so like trying to piece together a complex jigsaw puzzle where loads of the pieces look almost identical.
Exactly that analogy.
You could easily end up with gaps or put bits in the wrong place,
Hmm.
especially in those repetitive parts of the genome.
Precisely. This fragmentation could lead to significant errors during the assembly process. You end up with a skewed view, potentially, of the actual viral genome. It hindered our ability to fully grasp the true genetic variation.
That sounds like a really fundamental challenge. But technology moves on,
Yeah.
right? What about the newer third-generation sequencing platforms?
Absolutely. And this is where the story gets really compelling. Newer platforms like PacBio and Oxford Nanopore Technologies, ONT, brought something revolutionary to the table.
Long reads.
Long reads.
Okay.
Instead of tiny fragments, they generate much longer stretches of DNA sequence, sometimes thousands of bases long, that can span much bigger genomic regions.
So that sounds perfect for those tricky repetitive regions that confuse the short reads.
Exactly. It helps resolve those ambiguities.
I sense another but coming. There's always a trade-off.
There often is, yes. Let's take PacBio first. It can achieve remarkable read accuracy. Really high, sometimes up to like 99.997%. Incredible precision.
Wow.
However, for widespread use, it comes with a pretty hefty price tag, both for the instruments themselves and the cost per sample. The workflows are complex, you need specialized infrastructure, and it demands substantial computational power and data storage.
So super precise, but maybe not accessible for every lab or for large-scale surveillance.
That's generally been the case, yes. It's powerful, but comes with significant logistical and cost barriers.
Okay, so how does Oxford Nanopore Technologies, ONT, fit into this? You mentioned them too.
Right, ONT. It's really gained a lot of traction, partly because it offers a more accessible route. The sequencing cost is relatively lower.
Okay, the book less.
And crucially, its long reads are excellent at spanning those larger genomic repeats and complex structures that really trip up the short read methods. That capability is a significant advantage.
Right, so if ONT is more accessible and it handles those complex regions better with long reads, what's the catch there then? Because there usually is one.
A very good question. Historically, the main catch with ONT platforms was their inherently high error rate.
Ah, okay. The data wasn't as clean.
Exactly. These errors could pop up as spurious substitutions, insertions, or deletions. We call those indels, and single nucleotide changes, SNPs.
Right.
For researchers, these errors created a big hurdle. It made it really tough to accurately reconstruct the true biological genetic variation they were seeing. Distinguishing real variation from sequencing noise was hard.
So great for getting the length, but the raw data itself was a bit messy.
Noisy.
Precisely. Noisy is a good word for it.
Oh.
And adding to that challenge, there was a real lack of standardized, end-to-end pipelines specifically designed for ONT data.
So researchers had to kind of patch things together.
Pretty much. They had to find different tools for each step, from base calling, which is translating the raw signal into DNA letters, all the way through error correction, assembly. It wasn't streamlined.
This sounds like a bottleneck.
It was a major limiting factor for integrating this really promising technology into routine research and clinical work. Okay, so this brings us perfectly to the heart of our deep dive today, BonoboFlow. This sounds like it's positioned as the innovative solution designed to tackle exactly these persistent challenges with ONT data.
That's the idea.
Turning those historically noisy long reads into accurate, reliable viral genomes.
That's right. What's really clever about BonoboFlow, I think, is how it systematically addresses that messiness of the raw ONT reads.
How does it do that?
Well, it's a standardized pipeline built using NextFlow. And for listeners who might not know, NextFlow is a really powerful system for managing complex computational workflows reproducibly.
Okay.
BonoboFlow is specifically engineered to streamline the entire process for ONT viral genome assembly and, crucially, haplotype reconstruction.
Haplotype reconstruction, getting those different variants within a sample.
Exactly. So its core purpose is high accuracy base calling, robust error correction, proper genome assembly, and that detailed haplotype reconstruction. It even ensures the final output is reliable by correcting reading frame issues in the draft assemblies.
Reading frames. Making sure the genetic code is read in the right groups of 3.
Precisely. So the whole process becomes much more reliable and robust from start to finish.
Okay, that sounds incredibly comprehensive. Can you walk us through it, maybe step by step, like following a recipe to bake the perfect genetic cake? What are the key stages?
Okay, yeah, the recipe analogy works quite well. It's a very systematic, carefully orchestrated process.
So you start with your raw ingredients, the sequencing data.
Exactly. BonoboFlow takes either raw POD5 files, that's the direct signal output from the sequencer, or FSTQ files, which have already been base called into sequences.
Right.
If you give it the raw POD5 files, the first step is base calling. It uses a state-of-the-art tool called Dorado with a super high accurate, or SUP model. This translates those faint electrical signals into clear, readable DNA sequences.
Like decoding a secret message into plain text.
Kind of, yeah. Then, once you have clear sequences, it's all about cleaning and sorting.
Right, prepping the ingredients.
First, it uses a tool called Chopper. This trims off any unwanted adapter sequences, little bits of DNA added during the lab prep. Think of it as trimming the excess dough off your cake.
Makes sense.
Then, if you've sequenced multiple samples together in one run, which is common...
To save time and money.
Exactly, needs to sort them out. That's demultiplexing. It uses unique barcodes assigned to each sample to put the reads into the right bowl, so to speak.
Okay, sorted ingredients. But you don't want any impurities, right? Like host DNA getting mixed in with the virus.
Absolutely not. That's the next crucial step, contaminant filtering. It uses a reference genome, say, the human genome, if you're looking at a human sample, along with tools like Minimap2 and SAMtools. It intelligently identifies and removes reads that match the host or other known contaminants,
Sifting out the junk.
leaving you with just the desired viral reads, focusing on what's truly relevant.
Okay, so now you have pure sorted viral reads. But what about those historical ONT errors you mentioned, that noisy data? How does BonoboFlow deal with that?
Right, that's where robust error correction comes in. This is critical. BonoboFlow employs a sophisticated tool called VChat. Importantly, it's haplotype aware.
meaning it considers those different variants when correcting.
Yes, it tries not to accidentally correct real variation into a single consensus. It addresses those spurious substitutions, indels, SNPs, directly tackling ONT's historical weakness. It's like a very powerful spell checker and grammar corrector for the genetic text.
Fixing the typos.
Oh,
Okay, and then finally putting all those corrected clean pieces together.
yes, the assembly stage. Or haplotype construction, depending on what you need.
So it can do either.
Yes. If you want a single consensus genome representing the dominant strain, it can use an assembler like Fly. Or, and this is really powerful, if you need to resolve those individual viral variants, the haplotypes, it uses an optimized version of a tool called StrainLine.
So it can build one picture or multiple distinct pictures if different strains are present.
Exactly. That's where the genetic pieces finally come together coherently.
So the cake is assembled. Does it need like a final polish?
It does. After assembly, there's genome polishing. It uses another tool, Medica, to further refine the accuracy of the consensus sequence. It smooths out any remaining small errors, like spurious indels and SMPs.
Like adding the final glaze, making sure it's smooth and perfect?
A good analogy. And crucially, it includes that frame shift correction step we mentioned. Using tools like Diamond and ProofFrame, it checks the polished sequence for any frame shifts places where the reading frame might have slipped.
Right. Ensuring the genetic instructions are read correctly, making sure the sentences make sense.
Exactly. That's vital for predicting the correct proteins.
So the final output isn't just a sequence, it's a biologically meaningful, accurate blueprint, or blueprints, if there are multiple haplotypes.
Precisely. You get FASTA consensus files that authentically reflect the haplotypic diversity in your sample with corrected frame shifts, ready for downstream analysis.
And how practical is this for researchers in the real world? Is it easy to set up and run?
That's a key point. Its implementation using Docker and Singularity is a huge plus.
Containerization, right. Packages everything up.
Yes, it means scientists across different computing environments, big clusters, maybe even a powerful laptop, can deploy and run it relatively easily. It ensures reproducibility,
So more accessible.
much more accessible, and importantly, BonoboFlow is designed to work well with the latest improvements in ONT hardware. The newer R10 flow cells and Q20 Plus sequencing kits, they already offer much better raw accuracy.
Oh, so the input data is getting cleaner anyway.
Yes, often achieving an average Fred quality score around 30, which translates to over 99% base calling accuracy. BonoboFlow leverages these improvements, meaning its performance in real-world applications should be even better now.
Okay, it sounds incredibly comprehensive, well-designed. But in science, the proof is always in the pudding, right? How does it actually perform when tested?
Absolutely. The researchers put BonoboFlow through rigorous testing, both using simulated data, where you know the ground truth, and real-world viral data.
What do the simulations show?
They simulated sequencing reads from several different viruses: Zika, polio, adenovirus, HIV, even the large vaccinia virus. They simulated them with a fairly challenging 10% error rate, so 90% accuracy, and a decent sequencing depth of 100x.
And how did BonoboFlow do compared to other tools?
Well, BonoboFlow, along with some other established pipelines like StrainLine and Accuvir, generally performed very well. For many of the viruses like polio, HIV, adeno, Zika, they typically showed alignment percentages above 99%. Pretty good reconstruction.
High accuracy, then. Were there any key differences or limitations found?
Yes, some interesting points emerged. For instance, some of the benchmark tools like Accuvir and another called Genome Detective sometimes struggled to produce separate consensus sequences for all the individual strains put into the simulation. They often just generated one overall average consensus.
So they missed some of the diversity.
Potentially yes in those simulated scenarios. Also, the original StrainLine pipeline, which BonoboFlow optimizes, was noted to be limited by high memory usage. With very large data sets or really long genomes like adenovirus and vaccinia, it could sometimes fail.
Scalability issues.
Right, which raises questions about practicality for very complex or large viruses. Interestingly, BonoboFlow itself also showed some challenges reconstructing the really big and complex ones like vaccinia.
Oh, why was that?
The researchers suggested factors like highly repetitive regions in the vaccinia genome, and just the sheer computational demand made it difficult. It highlights that while BonoboFlow is powerful, there are still areas for future refinement, especially at the extremes of genome size and complexity.
That makes sense. Continuous improvement. Okay, so simulations are one thing. What about the ultimate test? Real-world samples. You mentioned they use HIV-1?
Yes, they turned to HIV-1, a really important test case because it's known for its huge genetic diversity within patients and obviously its clinical significance.
Right.
They generated long-read sequencing data using the ONT Minion platform, specifically with the newer R10 flow cell and Q20 Plus kit, so expecting good quality data from 24 archived HIV-1 plasma samples from Uganda.
And the data quality was good.
It was. Average read length around 4,700 base pairs, average Fred quality score of 30, so high bait calling accuracy going in. They then benchmarked BonoboFlow against Accuvir and Genome Detective using this real HIV data.
Okay, moment of truth. What were the critical findings?
Well, first off, the phylogenetic analysis looking at the evolutionary relationship showed high agreement. All the pipelines generated consensus sequences that clustered closely together. That's reassuring.
Good consistency.
Yes. BonoboFlow also showed strong sequence similarity to the others. Around 96% means similarity against Accuvir, and nearly 99.9% against Genome Detective median similarity. High agreement overall.
But was there something unique BonoboFlow did?
Yes. And this, I think, is the crucial insight, the real aha moment for listeners, perhaps. BonoboFlow demonstrated a unique capability. It identified more than one haplotype in some of those real HIV-1 samples.
So it picked up on that within patient diversity, while the others maybe just gave an average.
That seems to be the implication from the results presented. And for RNA viruses like HIV, which mutate incredibly fast and generate huge diversity, this is not just a technical win, it's potentially a game changer.
Why is finding those multiple haplotypes in one sample so important, especially for HIV?
Because it means we can finally start to map the full genetic picture within a single patient. We can identify those individual variants that might be driving drug resistance or influencing disease progression or how the virus is transmitted.
Instead of just getting a blurry average picture.
Exactly. You're moving from a generic overview to a high-resolution map of the actual viral population inside the host. This ability to capture intrahost diversity accurately is absolutely essential for better clinical management, for tracking resistance, for tailoring treatments. It leads to much more informed decisions.
That really highlights the value.
Okay.
Beyond accuracy, the other big practical thing for researchers is efficiency, tying in computer resources. How did BonoboFlow stack up there?
Yeah, computational efficiency is vital. You don't want a pipeline that takes weeks to run.
Right.
BonoboFlow processed a pretty substantial test file, 2.1 gigabytes of FSTQ data, over 770,000 HIV reads in just 1 hour, 3 minutes, and 21 seconds.
That sounds pretty fast for that amount of data and complexity.
It's certainly practical for research settings. They noted that the haplotype construction step took a significant chunk of that time, which makes sense. Resolving multiple similar variants is computationally intensive.
Understandable.
But what's also compelling is the flexibility. It ran well on high-performance Linux clusters, as you'd expect. But the paper also mentions successful runs on consumer-grade systems, like macOS and Ubuntu laptops.
Really? So it's accessible even for labs without massive computing infrastructure?
It appears so, yes. That makes it much more widely accessible to the scientific community, not just labs with huge budgets or supercomputers.
Okay, so let's zoom out. What does this all mean for the bigger picture? BonoboFlow isn't just another software tool. It sounds like it represents a significant step forward.
I think it does. It successfully seems to balance accuracy, computational efficiency, and that crucial accessibility. It provides a needed end-to-end solution specifically for taking raw ONT reads and turning them into polished, reliable consensus genomes and haplotypes.
Tackling those bottlenecks we talked about earlier.
Exactly. It directly addresses those challenges, the error rates, the lack of standardized pipelines that were identified in previous studies with nanopore tech and assembly tools. It contributes to refining the whole field.
And it's open source, right? Available on GitHub.
Yes, that's another big plus. Being open source means it's freely available. Anyone can use it, inspect the code, even contribute improvements. And importantly, unlike some online web tools that often have strict limits on how much data you can upload, BonoboFlow can handle larger data sets locally or on clusters.
Fostering collaboration and speeding up research.
Absolutely.
So thinking about the real-world impact, where do you see tools like this making the biggest difference?
Oh, the potential applications are huge. Think about virus surveillance. Being able to quickly and accurately sequence viruses from samples allows for rapid monitoring of new or circulating variants. That's critical for public health, for pandemic preparedness.
Spotting trouble early.
Exactly. Then there are phylogenetic studies. With more accurate haplotype-resolved genomes, researchers can build much more detailed evolutionary trees, track how viruses spread through populations, understand transmission clusters better.
Deeper insights into evolution and spread.
Yes. And it just enables more advanced viral characterization overall, performing multiple sequence alignments, accurately estimating open reading frames for protein prediction, doing subtyping or genotyping, all become more reliable, leading to a much deeper understanding of viral biology, from a single infection right up to global patterns.
It sounds incredibly powerful, that ability to see so much more detail and get it relatively quickly.
Indeed. And as we mentioned, the underlying ONT technology keeps improving. That R10 chemistry, the Q20 plus kits, they're already giving roughly 99% raw read accuracy. As that gets even better.
BonoboFlow's job gets easier, and the results even more reliable.
Exactly. Its performance, especially for maybe rapid field applications, is likely to improve even further. Now, are there still challenges? Yes. The paper acknowledges that things like variant calling in really, really long genomes with lots of complex repeats could still be improved, but it's a substantial step forward.
So wrapping up our deep dive today, we've explored BonoboFlow, and it really stands out as a brilliant example of how these targeted computational tools are revolutionizing our ability to understand that invisible world of viruses, making that shape-shifting entity maybe a little less mysterious.
It really is about moving beyond just identifying a virus. It's about truly understanding its intricate genetic landscape, its evolutionary tricks, its potential impact, whether that's on human health, animal health, or even environmental surveillance. That kind of critical, detailed analysis is just essential in today's world, especially with the sheer volume of information we face.
Absolutely. So a final thought for you, our listeners. The next time you hear about a new viral variant emerging or a sudden outbreak, take a moment to consider the incredibly complex detective work happening behind the scenes. Think about tools like BonoboFlow, meticulously reassembling these tiny genetic puzzles, often revealing not just one picture but several crucial variations within a single sample, helping us see the unseen with breathtaking detail. And maybe ask yourself, with this level of genomic insight becoming faster and more accessible, what new frontiers does this open up? Could it truly revolutionize disease prevention, lead to genuinely personalized antiviral therapies, or even help us predict and perhaps prepare for the next major viral threat looming on the horizon? Something to think about.